You're asking about the somatic patterns of life. Basically, are you familiar with Chladni plates?
No.
Basically, they have particles like sand on a thin metal plate, and they resonate those plates with frequencies such as notes or things like that, and it forms shapes. And those shapes are remarkably similar to various structures, and also, so basically, vibrational frequencies, electromagnetic frequencies, carry information or organization at certain frequencies, and they produce certain shapes. And this is just showing some similarities of patterns, three-dimensional patterns associated with vibrational that translate into living structures. As the earlier book I was talking about, Fields of the Cell, cover this in depth. And for clarification on that, I would highly recommend that, because that's going to give you your morphogenic field ideas, concepts, and how this is integrated and why it is important. That's the whole basis, is why we need to look at that, is the importance here. And the next, actually the last third of this, which is what's basically left, is really going to deal with these the frequencies and why they're important and how they affect things. So probably be best if we go on there. But exogenous, external, endogenous, internal. Okay.
Well, let me just say
with cells and molecules and structures in living things, either constructively, which would be coherently, or destructively, decoherently.
So is this kind of going along with your the description of the endogenous structures would be the microtubules and how the cell is set up and the communication between cells, correct? Just to follow?
Yes. Well, internal structures are endogenous, but we're talking about that in relationship to electromagnetic fields. So we have electromagnetic fields within us. They're operating. We have the soliton. We have these eigenfrequency, or what is called eigenstates, that are a result of these chemical processes and electronic and electromagnetic processes going on within our cells. We talked about that with the Hofstadter's butterfly and how that related to cavities and the 12 fractals that made up the patterns that actually led to EEGs when we're talking with the brain. Okay, so those are endogenous electromagnetic phonons, magnetic fields that are within us that regulate how our body operates. Then there's exogenous EM fields, such as using the laser, or transcutaneous magnetic stimulation, okay, or electromagnetic, transcutaneous electrical transcranial electro stimulation. Those are all exogenous forms, or being exposed to your Wi-Fi. That's an exogenous electromagnetic field. It affects us, okay? What we're trying to do is, as I said, take that carrier wave and modulate it with additional frequencies that are beneficial to structures in the body. So that's what we're working, the idea and why this might be important, and that's what we're going to really go into. And you know, this slide here kind of covers some of the things we ought to be looking at, along with that previous slide that you noticed that we're not doing, and we talked about some that we're only doing part of. But here are the things that we need to also consider as part of the importance of the different frequencies, the frequencies that are going to resonate beneficially with our cells, increase the coherence and the constructive activity, versus destroying that coherence and potentially causing some form of dysfunction or disease. That's what we, you know, that's the whole basis of what we're talking about here. That makes sense?
Yes, sir.
Okay. Now I'm kind of looking over you, not the camera, so I hope I'm not too ignoring the audience when you go there. But yeah, we can read these. You know, electromagnetic fields directly influence the transmembrane ion gradients, such as calcium channels. Okay. Calcium is extremely important in the cellular mechanisms. It's involved in almost everything. But the next thing, you know, the specific electromagnetic frequencies can cause vibrational resonance in macromolecules. We talked about that. The cells, and including DNA, DNA, the nucleotides, ion channel proteins, microtubule proteins, cytosolic proteins, apart from their chemical signaling, and they may communicate through their vibrational character, okay, which we talked about also. Cyclic polarized EM fields can penetrate life's tissue to promote coherent vibration in various cell components. There has been a conversation or discussion about whether polarity was important to light. Apparently it is when you look at this. That's one of the considerations that may lead some credence that we might need to have polarized light, okay, and use that rather than, you know, non-polarized light. Extended fields of coherent molecules were assumed to be formed by a process called Bose-Einstein condensation. We don't want you to get into that sophisticated stuff. It just is basically that quantum processes can occur at you know, the temperatures, the wet, noisy, warm environment of the body, and they can't be discounted and say, oh, they just don't exist. Electromagnetic phonon, electron wave packets can be converted to mixed forms. We talked about that, the positrons, solitons, and the polaritons, okay, which are the photon electron.
They have a name for that. That's important.
And we talked about the solitons in the cells being able to constitute a part of the local quantum field that can be involved in the intracellular morphogenic ordering or structure, if you want to call it that, as well as intracellular communication. And we talked about it. It was instrumental in the folding of proteins, you know, native and non-native aggregation. Long-lived rotational states of some molecules in protein structures, spinning magnetic moments and radical pairs, endogenous cellular magnetic nanoparticles, and even magnetic moments of protons in liquid water are all possible targets of magnetic field interactions. And we have that with laser light, well, actually all light, okay? All right. So the question that everybody has, and we touched on before, is what are eigenfrequencies? They are the natural frequencies of the system, okay? And its various subcomponents. And they form what's called eigenstates, natural states, which are basically of the resonance in the biomolecules and cells, okay?
Got it?
All right, and they tend to oscillate in the absence of any driving or damping force. Okay, so
follow.
And here's an example, and I can't blow this up a lot larger, but they can copy the picture and do it so you can read the frequencies. But these are the normalized 12 frequencies, and those little dots above are a summation of how well this fractal pattern, these 12 octaves, which are basically based on very similar to musical notes, classical notes that were there, and how accurate they are as either positive coherent effects, beneficial effects in cells, or there's another slide that follows on decoherent or pathological frequencies that cause problems in cells in living systems. Okay, and the dots, basically they've taken like laser light or electromagnetic frequency or electrical frequencies, and they plotted this, and you can see how well they correlate with these 12 fractals or these 12 eigenfrequency bases.
May I point out one thing here that I see? What I can see there is that none of those are above 1,000 hertz. Why is that? Because it's algorithmic? Right there's like 460 is the highest I see, something like that.
Yeah, exactly. But what they did is they reduced it down into these graphs. So they didn't give you a list of bazillion or thousands of frequencies here that are doing stuff. What they do is they gave you the formula, but they reduced it down to some to a certain frequency spectrum.
Numerical. It's based on these different algorithmic frequencies. These are the building blocks of the rest of them, correct?
This is basically a normalized, it states a normalized mathematic acoustic reference scale. It's a reference scale at hertz frequencies.
Gotcha.
Okay, but as it says here, it's from 97 different frequency data from 172 biological studies at the hertz, kilohertz, megahertz, gigahertz, terahertz, and picohertz frequencies. So it's been reduced down, but the point being is you see how well they align within the same, I guess, channels.
Packages or channels of those things, right?
It's a very narrow band. That's why I said at 8 hertz, beneficial effects. At 12 hertz, not beneficial effects. That's how close they can be. And that's why it's important to be able to have instruments and have our lasers be able to be tuned to what, to be able to research and take information that's out there and make some therapeutic sense out of it. Does that make any sense to you?
I'm following.
Yeah.
Click.
You said yeah, but or maybe I don't.
Go ahead. I'm following.
Oh, okay. I'm sorry. All right. Basically, again, coherent EM frequencies promote coherences and increase the strength and fidelity of the intracellular signaling or the soliton. And decoherents do exactly the opposite. And they've done this work and they've shown these effects of coherent versus decoherent. Not the same as coherent light versus non-coherent light. Doesn't mean non-coherent light can't have it in the proper sequencing, could not be biologically coherent. Does that make sense?
I got you.
Okay. So look at this. The stabilizing electric fields observed with coherent electromagnetic frequencies, therapeutic nerve stimuli. The ones in yellow basically are things that directly or easily can be associated with transcranial photobiostimulation therapy. Therapeutic neurostimulation, therapeutic brain stimulation, therapeutic special. You can read these. I know everybody's probably gone through most of them. And then on the other side, we have when you have decoherent electromagnetic frequencies that disrupt these endogenous eigenstates, then you're going to have cells dysfunction because they're not communicating. It's like, imagine how irritating it is when you turn on your radio or you're trying to focus on something and there's all this static. Okay, if you're trying to listen to somebody, you're missing half their words because all the static is distracting. Okay, and that's maybe an oversimplification, but a little analogy. Getting rid of this static in here, if you have decoherent electromagnetic frequencies. Now, if they're strong enough, they'll total wipe out the signal and no information gets passed. And of course, then you start this process of entropy, and energy is being dissipated rather than being utilized for biological work.
Amen. I got you.
Right, here's showing again, basically, these a mathematical plotting of a bunch of studies showing beneficial and negative or constructive or deconstructive effects in biological systems based on coherent or decoherent. Obviously, you can see the red is the decoherent green, and then the yellow is the transition states, which Kind of depends on different things. But the point on the thing is this is our basis for research on why they're important. We know that bad frequencies cause problems. Good frequencies help. And so we need to focus modulating our lasers to these beneficial frequencies, but even more specifically tied to the appropriate frequencies for the tissues that we're trying to treat. And then we can add in the other parameters on what's the best form of light to use to do it. Sometimes what we want to do is boost cell water, okay? The 980, 1064, even infrared is fantastic at cell water ordering. That water, interfacial water surrounds all proteins, all structures inside the cell. Perfect example, our body is, what do you say the percentage of water is in the body?
90%.
You know, yeah, it's getting up there. All right, if I give you a steak, I'll buy the dinner. You can squeeze a drop of water out of it. Can't do it.
Well bound. It's well bound.
It's bound. Yeah, well, you know, you're right. It actually forms a gel clathrate, okay, a polymer. Okay, it is not free bulk water. It's not the water, you know, in your water bottle or, you know, a glass of water or something like that. We're not bags of ionic fluid, okay, bound by this cell membrane that's keeping some stuff out and some stuff in, okay? And if we puncture it, it's just going to leak out. That's not how it works, okay? And that was one of the things that Ling was pointing out to on, you know, on the cell wall. He showed where he shaved off what is supposed to have been the cell wall, and the cell functioned just fine, okay? So those are side things on it, but the, you know, the importance is when we do things, we need to do it for a purpose and target our therapies that way to intelligently apply our photobiostimulation. That's really...
I got you. Let me summarize it. So not only do we have to, this is just an expansion of this, pick out what wavelength or color of light, what power, you know, what dose. Then if we pulse it at a certain frequency of hertz, will influence different molecules and structures and organs and tissues of the body in a positive way or in a negative way. So those things have to be paid attention to. That's what I'm getting, right?
Yeah, that's pretty close to it. Except for, we'll get a little bit later, there's other ways to modulate that light frequency.
Okay.
Pulsing is one, and maybe perhaps the easiest. Okay, and we'll talk about this a little bit later because otherwise I won't have anything to talk about when I get there.
Oh no, go ahead.
So here's some data on resonant frequencies. Okay, now you can see, you know, you were talking before about wavelength. Okay, and here's your hertz. Okay, and you probably don't, if you don't recognize, you know, the scientific notation of various things, let's go down to a frequency that everybody recognizes. Okay, for example, okay, here's, okay, I went too far. What did I do with it? There it is. There's 851. That's basically 3.52 times 10 to the 14th, okay, or 352 terahertz. Okay, we know 810 is around 370 terahertz. Okay, so we're getting, as our wavelengths get longer, the frequencies start decreasing, right?
You tell me. I failed the counting. So the more numbers you put up, the farther away I get from understanding it. That's fine.
Yeah, I can barely hear you now, Joe.
I said that I failed the counting. So the more numbers, you put up a column of numbers and and I'm going to have trouble. So
Okay.
I see this 851 nanometers, and that is converted to a hertz that I've never seen done before.
Okay, but you'll notice as our wavelength gets longer, okay, as we go from left to right. Normally when we look at the electromagnetic scale, the frequency drops, right? You can see that. You know, the 715 is you know, 4.1, 419 terahertz, and we get to 851, we're down to 352. When we get up to okay, there's close to the 1064, we're down to 280 terahertz. Okay, as far as frequency. Okay, so it's becoming, the frequency becomes lower and lower and lower. Okay, so now whether we need to modulate at that level or to get the frequencies at that level, if those are eigen, you know, as eigen frequencies, there probably are fractals of that in that transference below that that are harmonious with that, that can add to the hertz frequency based on that algorithm that we were looking at the previous page. Make sense? I noticed that one of the prior authors was, and just to bring this up, but they were using green light in, I think it was it 532, 33, okay, and proteases. And they were talking about bone healing of fractures, okay? Well, one thing it doesn't tell you about the proteases here is there's different types of proteases. Some of them are inhibited and some of them are increased or go about their function, but proteases break down protein, okay? And in some cases, certain proteins that break down are beneficial to bone healing. In some cases, they inhibit it. So we don't know there, but in this case, that green light should have been beneficial, but it wasn't. It didn't do anything, where the 810 light did stimulate the healing of the bone. So that's the importance of these frequencies and understanding stuff. And we were just using that light, okay? We weren't modulating or doing anything else. So it gets a little complicated. I'm saying this is something that people are going to just do tomorrow, but we need to be thinking in this direction because it has biological effects that we can take advantage of.
What you're pointing out here is that the protease 533 on the far right scale is a decoherent frequency.
No, it's a coherent, it's a protease inhibitor right there. On this one was 565.
Right.
Okay, but which proteases? Doesn't specify. It's a class of things, and proteases do what I said. They cleave proteins and they open that stuff up. So there's purposes in that. In this particular research paper, so when you look at research, you have to, you know, the devil's always in the details, and you have to look at that and say, okay, why didn't that work? We can't just assume that every you know, green light doesn't, you know, help with bone healing.
I understand my point. My question was on the far right column, you have coherent as a 1 and decoherent as a 0, the far right column.
Oh, yeah. Okay. Yes. And in that case, on those proteases, it was decoherent. Yes. Okay. And so that would be consistent with what they experienced, and it didn't do its bone. But I'm saying that there are also reports on proteases stimulating bone healing, and they're using it for that. But there's different classes of proteases, okay? So his result, he could have looked at this thing and said, I know that 533 probably isn't going to work if he knew which protease it was, but in chances are he shouldn't have been surprised, okay?
I follow.
All right. Okay, let's see. and the same thing with the 851 light. That isn't necessarily always a good coherent frequency. Unfortunately, that's what Chattanooga manufactures their their lasers at. Okay. So yeah, you're right, but okay. Well, everybody's got the ideas on what's going on here, which is the important part. Okay. Microtubules make up the cytoskeleton of cells, and they do also of neurons. So neurodegenerative diseases are basically might be classified as microtubule diseases. Now, that might be an oversimplification, but there's certainly that component there, okay? And we know that microtubules are responsible for electromagnetic photon-phonon guided information exchange, as well as neuronal morphology, ion channel function, protein function, and intracellular transport, which is what we've been talking about all the time in all the studies. Okay, so the central question is, how do we deliver these eigenvalent frequencies and quantum information restore coherency or coherent vibrations in the CNS neurons? And importantly, they're microtubules to produce a return to optimal healthy function. And here's where we come in. As I've talked about before, is lasers or NIR light makes an excellent carrier wave. And like I said, fiber optic communication has utilized this for years now. And how they do it, they modulate either amplitude or phase modulation or frequency modulation. Okay, so here's just some examples on how they do it. There's a frequency, there's a carrier wave frequency, and they can modulate that either with amplitude, AM, that's AM radio, and that's FM radio. Okay, frequency modulated and amplitude modulated. Okay, this may be a little more complicated, but next slide or one of the following slides is going to talk about polychromatic light. I don't know if our diodes presently technically are capable of doing that. That's a less interesting, I think that might be much harder to do. And certainly probably I can't answer the technical questions on that. Amplitude modulation might be the next thing over pulsing changes. And of course, this everybody recognizes, that's the pulsing process that we're using in lasers, the super pulsed and the usual pulsing that,
If I could, this is there's a lot of information on this panel here, but one of the things is that a pulse is going to be on and off, but then there's also what they say is the duty cycle, which changes it also. And can light be considered on off as far as a square wave pulse, or is it more sinusoidal? So this might be a little bit too technical, but if it is, you're talking about pulsing on, there's like freaking 3, 4 different, 5, who knows how many models you're showing here.
Okay. Well, the wave itself is not going to be a square wave. Okay. But on and off certainly could be, as shown there, as being square, as the wave light itself is going to be sinusoidal in shape, pretty sure. But the presence of it going on would be basically more like, if your on-off cycle would be like this. Okay, when you turn it on, boom, you got this frequency pulse, and when you shut it off, it's not there. Okay,
got it.
It's not going to go up. There may be some ramp up time, but the speed of light is so fast enough for practical purposes. It's instantaneous as far as at a macro scale that we're concerned about. So it's not going to be a big rounded thing. Now, the penetration into tissues, kind of, that there may be somebody that can say that, the penumbra and things like that may contribute somewhat on as far as concentration at depth, density at depth. Okay.
Got it.
All right. The other thing would be to use polychromatic, in other words, combination light therapy, which is already being used, and then be able to modulate those or pulse those at different rates. Okay, but we can't do that now with the current equipment. Okay. And I've showed you this before. This is the various things that show up on the IR spectra, and it starts at 1,000. Okay, but point being is that different frequencies can, if you had a couple of these frequencies and you want to use those to hit certain structures and affect certain molecules, because here's all the different molecules that can be affected. And there's obviously all of them can be, but it's just, you get the big picture here, that if you wanted to treat
so if you look at it, I got
more than one molecule or structure, we might need to use different frequencies, and we could use and even it could even be theoretically the same light, a same laser frequency modulated or pulsed at a frequency that is designed to affect that tissue more than any other tissues.
I follow you.
If it doesn't match up in the frequency, it's passing through anyway.
I follow you. So let me make an analogy and see if I'm close. Okay. What if this was a 12-string guitar or a 24-string guitar, and this is where you were putting your fingers when you strummed it to get a good sound or a discordant, dissonant sound?
At first blush, that sounds like a good analogy. I hadn't thought of it, and we talked about musical instruments. If, the one thing, and Giesing and Meyer does talk about that, is notes, certain notes, it's, if you just put your fingers on a guitar and you strum the strings of a guitar, and you've got notes that aren't in harmony or on the same fractal scales as the other ones, it's going to sound like crap, and you don't do it. But when you have the notes right, the fifth, the third, and they're following the patterns, it sounds great. Just like an orchestra.
Coherence.
You're right. I think that's a good analogy to maybe explain how we need to think about how we could produce more effective transcutaneal photobiostimulation, or affect any therapeutic laser apparatus. Okay. All right.
I am the moderator.
Yeah. Go ahead.
No, I am the moderator. Oop, go ahead. Go ahead.
Oh, I thought you had something else you wanted to add in there, another question. So we're getting to the end here. There is a field of science called optogenics, okay? And that's basically, they use what is normally called upconversion nanoparticles. And I think we talked a little bit about this at the beginning of, prior to starting our presentation with you, where, for example, carbon-carbon double bonds, okay, molecules generally respond well to ultraviolet light, okay, class of frequencies 270 comes to mind, 280 around there, and then lower down in 220-ish or something like that. Depends upon what adjacent bonds and what structures are also part of, but they tend to be in that ultraviolet light category. When you have these upconversion nanoparticles, they actually can absorb and be responsive in our light, but when those electrons are excited and jump to the higher level and they come back down, they release an ultraviolet bioluminescence. And that can be used because ultraviolet light generally runs into enough energy to eject an electron or stimulate a free radical activity. Okay, and if to inject that or couple them to nanoparticles that localize to a particular, say, a tumor, that's drawn to using monoclonal antibodies that attach to a tumor, hit it with, your NIR light, it upregulates and basically uses ultraviolet to destroy the tumor, or a virus or something similar to that. Okay, so that's what's going on there. That's another possible way to modulate known light frequencies using NIR light. Okay, it's a little bit different than what I was talking about, modulating the frequencies. And people are already looking at doing this type of thing, and I just thought I'd throw it in there so people would be thinking about it, because my research in nanomedicine involves the use of nanocarbon fullerenes, and they're relatively insensitive to NIR light, but they love ultraviolet light. And ways to stimulate them with the ultraviolet light, they become very potent and toxic to cancer cells. So, there's a series of steps, but it's interesting. I just thought I'd throw that in there for something exciting, because I know there's medical doctors here, and some of their compatriots may actually be involved in some of this stuff. So
I got you.
The thing that's important is frequency matters in biological systems, and really, as we talked about, alluded to, is what we observe isn't nature. Okay, the questions we ask of nature are what nature reveals to us, and we need to start asking better questions.
I got you. Well, thank you, Doc. Let's go up. Let's go up a little bit and just let me ask a question. Even on go up one slide, if you would, because
I'm going to have to lean in. I hope I don't get too big on the screen, but I can barely hear you.
Go up one slide just for the last question. There we go. So you're saying here, the development of near-infrared light-activated particles optogenetically have greatly extended the application of light for neuromodulation in deep tissues. Reading the second paragraph. My question is, don't we have bacterium and cells within our body that will bioluminesce when we hit them with the light? Don't we already have some kind of endogenous nanoparticles, if you will, nanomaterials?
Yes, and there's a good discussion of that in fields of the cell, but the technical name for them is ultra-weak photonic emissions. And it depends upon the structures involved and things. You can luminesce different ways. Certain bacteria luminesce in the 400 and 500 wavelengths. Okay. Generally, in our cells, one of the biggest things, which is easy, everybody knows about it. Everybody knows about infrared scopes. We're producing that.
Infrared what?
Scopes.
Oh, to look at people.
The military all the time, they fly around. They got FLIRs on the police helicopters looking for the guys running out through the woods. That's those ones where if very common in there. Our bodies, produce heat, but how is heat produced? Okay, we know about generally that is a result of our chemical processes are associated with energy production and actually, believe it or not, free radical production. Okay. Interestingly enough, one of the adaptations that we did in an evolutionary process was uncoupling proteins, according to the current theory anyway, that what they're doing is they're allowing the inner mitochondrial membrane membrane to leak protons, so there wasn't as much force driving the ATPase.
Okay.
And creating ATP. Okay. If you don't, one of the things that is important that we may be doing, and it may actually reduce the free radical production of the body, is in people that have an excess of electrons from their food. In other words, they're overeating and they don't have the caloric demand. Okay. The mitochondria are inhibited by excess ATP. So if you're not using up your ATP, then you're shutting down the ATPase. However, you're still pumping electrons into your electron transport chain. Okay, uncoupling protons allow those electrons to continue to flow, because part of that process is exchange of hydrogen, right? We all remember that from our biochemistry. Okay, but as long as you're not letting that membrane differential get too high and drive that ATP thing that you don't need, because the body's inhibiting that, those electrons, if can continue to flow, and basic electron can transport chain can cycle, and that produces heat. And that's what we have in people in northern climes. They're generally a lot more efficient at dealing with cold weather, because they have evolutionarily developed to have uncoupling proteins. We have that in our brown fat, etc.
Okay, those are all key elements. But let me ask you this, because when you're talking about that, like the ion cloud and the things that you were talking about, I hear you saying that the light energy, the electromagnetic energy of light, can have a certain vibrational sense within the tissue. But a lot of people are talking about that the light energy is having an effect even on the outside, on the energy body, before it ever gets to the tissue. How does that kind of play into the things that we're talking about? Because I just want to know where you draw the line on the way that the light can be influencing this humanoid.
Well, from a theoretical basis, those are very interesting questions. I didn't want to get into this. I was going to throw it in because I'm already, it's already been long. Okay, but you know what a toroid is?
Toroid, nope. Is that 7 figures? Seven?
No, it is a. All right. You've seen pictures of these 360 degree waves that come out from a body in the heart?
Yeah.
Around. Okay, that's a toroid. Okay.
Toroid. Okay.
Toroid. Toroid. And so those are electromagnetic and magnetic fields. Electrofields. Okay. And certainly that exists from you as an individual, but also it's got to be, that's what I'm saying is all these places in molecules and the cell system and everything have this summation, this fractal pattern, but they all make up the toroid of the body. So if you're introducing a frequency inside that magnetic field, it's going to affect the whole magnetic field. Okay, weak magnetic fields from other planets are, they affect the tides, and they affect animal life and things like that. So yes, that is in there, but how at this point do we measure that type of thing? Okay, and how effective is maybe just delivering that type of thing may be therapeutic in itself. However, how do you deliver that weak thing without getting next to the body and getting it onto the tissues, considering we're not in a vacuum. That's not the only electromagnetic field that you're inducing to that toroid. It may be of not enough consequence. I don't know. That's a good theoretical question for discussion. Going back to your last question, I never did finish that and say, when we do laser 810 laser light and we stimulate cytochrome 4 COX, okay, we are turning back on without having protein uncoupling. And therefore, the important thing is that they found out is one of the biggest contributors to free radicals in mitochondria are the excess electrons in the electron transport enzymes. In other words, there's too many there and they just leak out. And there's always something there for them to interact with, the oxygen, and we get the superoxide anion. Okay, so if we can keep electrons flowing through there, we can hold down the free radical production. And say 810 or 808 laser light may do exactly that. I think 632 is also very effective at hitting that COX and keeping those electrons going, but you probably can't penetrate it deep enough, to get into a deep structure. So you're left with basically 810. That makes sense?
Gotcha.
That was where I was going. I just didn't get there fast enough for you.
So, when we get to the 2030 brain and transcranial photobiomodulation virtual summit in the future, what do you see our applications and such? How do you see it playing out 10 years from now, Doc?
Well, I guess it depends on what direction that we take. I think if we don't address at least some of the things I'm talking about, the importance of certain frequencies and things, we're not going to be much further ahead than we are now, because we have a given set of frequencies to work with in near infrared, right?
Right.
That's it. What are we going to do with those? We already got everything from LED to 60-watt lasers, which is far more than is needed to do transcranial work. Okay, and we certainly don't want to produce excess heat. So we have to look and expand the questions we ask so we can find more ways. We may find out when we do this, that's not so good, or we can only do so much of this because we just don't have the technology. Then the question goes to people that can actually have the tech, be the technicians and create this stuff, what they can do. So, I can't predict the weather tomorrow. So you asking me that in 10 years from now? It's a crapshoot.
How about the eternal sun? Is that coming up tomorrow?
The what is?
The sun?
Well, human beings are eternally hopeful, so eternally optimistic, because really we got one, there's a cognitive dissonance of the fact that we are fragile creatures on the verge of death at any given moment, and yet we continue to act like we're going to live forever. So what the heck? And as a former Marine pilot, there was a period of time where death meant nothing. It was for some other person. Unlucky.
That's why they don't send 40-year-old men into battle, or people my age into battle, because they're like, hey, those are real bullets.
Yeah. You want me to do what?
In the what now? Thank you, Doc. I appreciate your expertise. I appreciate the time you spent with us.
Hey, thank you for putting it together. I hope what I offered, didn't bore people. So.
So